Hello students, welcome to EPG Pathshala. I am Dr. Kalpana Bhargava from DEPAS DRDO. Today we are going to discuss about a module which is Opioid Peptide Biotherapeutics, which is under paper Molecular Therapeutics. So the objective of this module is to provide in-depth knowledge about the opioid peptide, their receptors and their therapeutic use in medical as well as in the research field. The emphasis of this module will be on the natural, semi-synthetic and synthetic form of opioid peptides and their use as biotherapeutics. Now let us see what is actually opioids. Opioids, as you can see the structure of morphine. Morphine is one of the most important and one of the most sought opioid. Opioid is a class of drug that act primarily on the body's opioid receptors. There are four different kind of opioid receptors. They are mu, kappa, sigma and delta receptors, and all these opioid peptides or opioid molecules, they act on receptors. So the most opioid receptors are found in the central nervous system as well as in the gastrointestinal tract. These opioids are usually primarily used for their analgesic effect, but also sometimes they are used for as a cough suppressant because it has got cough suppressant property. They are also referred, most often they are also referred as narcotics. So you might have heard of the term narcotics. So narcotics is nothing but they are the opioid peptides or opioid molecules. The definition of opioid peptide is any natural or synthetic compound that imitates properties of natural narcotics is actually called opioid. So what is the property of opioid peptide? It's an analgesic that works for binding to opioid receptors, which are also found principally in the central nervous system and the gastrointestinal tract. So these receptors mediate both the beneficial effect as well as the undesirable side effects. So these opioid peptides are peptides. They bind to the opioid receptors in the brain. Opiates and opioids mimic the effect of these peptides. Now, what is the origin of these peptides? The origin of these peptides may be produced by the body itself, which is actually called the natural peptide. For example, endorphins. The effect of these peptides may vary, but they all resemble those of the opiates. So, look at the classification of opioid peptides. There are different kind of opioid peptides. Number one, they are natural opiates. What are natural opiates? As the name says, these are the alkaloids and they are contained in the resin of the opium or poppy, morphine, codeine, thebaine. These are the natural occurring opiates. Then there are the second type, which are semi-synthetic opiates. So these semi-synthetic opiates are actually derivatives of natural opiates, which are modified based on their requirement and based on their activity. So as I said, they are created from the natural opioids, and some of the semi-synthetic opiates are hydromorphine, hydrocodone, oxycodone, and so on and so forth. Then there are third class of opioid peptides. These are fully synthetic opioids. These fully synthetic opioids are actually synthesized completely from chemical compound. So they are not the derivative of the natural peptides. They are actually synthesized chemically, and some of the well-known and well-established fully synthetic opioid peptides are fentanyl, pethidine, methadone. These are some of the most widely used and most commonly used synthetic opioid peptides. Then there are two types of opioid peptides. One is called exogenous and the other one is called endogenous. So as the name says, endogenous is the opioid peptides which are actually synthesized within the body and they are produced naturally in the form of natural peptide. They are produced in the body, and some of them which I will be discussing in my further slides, and some of them are endorphins, enkephalins, dynorphins and endomorphins. These are the peptides which are actually physiologically synthesized within the body. And then, as I mentioned, there is another form which is called exogenous peptide. Exogenous peptides are nothing but they are semi-synthetic and they are fully synthetic opioid peptides. So semi-synthetic, which are derived from the natural peptide, and synthetic peptides which are completely synthesized by using chemical compounds. First of all, let us talk about the endogenous opioid peptides. So endogenous opioid peptides, as I said, one of the endogenous opioid peptides are enkephalins and beta endorphins. So basically what all properties they have: analgesic, they have euphoria, antidepressant, reduction in anxiety. These are endogenous opioid peptides from important stress regulating system in brain. Now, what are the mechanism of opioid? As I think I already mentioned, that there are few receptors which are called opioid receptors, and these opioid receptors are actually receptors where these opioid peptides actually attack or work on. So, what is the mechanism of action for opioid peptide? So, what happens? It activates mu, delta, kappa, and opioid receptors. Opioid receptors are member of also G protein coupled receptor superfamily, and there are almost more than 1,000 members. The activation of these opioid receptors, it inhibits the neuronal activity, and that is how it reduces the pain. So why do we need to understand the opioid peptides? Because it offers more promise than the opiate in drug development. Because, and as you know, what is opiate? Opiate is anything which resembles to opium, which is a product of the opium, and which is actually originating from the poppy or opium. So now opioid peptides, these compounds are endogenous. As I said, these compounds are endogenous because they are synthesized within the body and they are actually part of the physiological system. And on metabolic degradation, they break down to amino acid. And since these amino acids are natural amino acids, they do not have any toxic effect into the body. The next point is that since these peptides are made up of several subunits, these amino acid residues, a large number of analogs can be synthesized. Also, these syntheses can have different modifications, can have desired activities, and can have many more factors and many more manipulations in their sequence in order to obtain the desired active peptide. Then these peptide analogs, unlike the opiates, are very flexible conformationally. As you know that conformation of any peptide or any drug plays a very important role in the activity of the drug or in any activity of the peptide analog. And since these are very flexible, they are supposed to be the ideal molecule to investigate the relationship between 3D structure and biological activity. In other words, we can call it quality and activity relationship. So now there are various technologies available for synthesis of opioid peptide, and these syntheses, as I said, there are many of them are available, but most common are the solution phase peptide synthesis and solid phase peptide synthesis. In solution phase peptide synthesis, these peptides are synthesized within the solution with different kind of activator, amino acids, and additive. However, in case of the solid phase peptide synthesis, these peptides are actually attached on a solid base resin, and one by one amino acid is attached, and that is how a sequence of peptide is formed. And this is the most widely used method in today's peptide synthesis, and which is very important as well as it is widely used method. To date, what we have observed is that these are the compounds, these are highly selective for receptors, and these compounds have been opioid peptides. They are supposed to be the very selective and very specific for any receptors. So, looking at the endogenous peptide produced by the body, as I already said that these are produced within the body and within the system, physiological system. They are again, as I said, they are opiate-like activity. They have opiate-like activity, and there are three major classes currently. And if you classify these peptides, these are endorphins, enkephalins, dynorphins, and endomorphins. Each of these families derived from different precursors. So, in other words, each of these endogenous opioid peptides have different precursors but has a characteristics anatomical distribution. So in other word, I can say that they are situated or they are located in different part of the body, but they have almost similar kind of activity and they have different kind of precursors. Now, as I discussed, there are many types of endogenous peptides. So the most important one is the endorphins. As I said, there are endorphins which are of many types, but the most important one is the beta endorphins. So any hormone which is secreted within the brain and nervous system and having a number of physiological function, they are peptides which activate the body's opiate receptor, causing an analgesic effect. As you can see that they are a 31 amino acid, the original endorphin is 31 amino acid residue peptide, and they form a very important class of endogenous peptide. The next one is enkephalins. Enkephalins are of two types. It is metenkephalin and leuenkephalin. So let us first discuss the metenkephalin. Why do we call it metenkephalins? It is called metenkephalins only because in the fifth position of the sequence, amino acid sequence, the sequence actually has methionine, and this methionine also, metenkephalin, also known as metenkephalin, which is INN. Sometimes it is also referred to as the opioid growth factor, OGF, and it is a naturally occurring indigenous octapeptide and has opioid effect of a relatively short duration. So as I mentioned, there are two types of enkephalins. I already discussed about the metenkephalins. Now I'm coming to the leuenkephalins. Leuenkephalins again has the similar sequence, except that the last residue in the sequence is the leucine residue. And as the name says, it is an endogenous peptide and it is supposed to be a neurotransmitter with the amino acid sequence, which is shown here. It is found naturally in the brain of many animals, including humans. The tyrosine residue at position 1 is supposed to be the analogous to the 3-hydroxyl group on morphine. Leuenkephalins has agonistic action at both of the mu as well as delta opioid receptors, which significantly greater preference for the latter. It has little to the effect on the K opioid receptors. Next class of the endogenous peptide is Dynorphin. Important class of Dynorphins is actually Dynorphin A. As you can see in the structure, it is actually long residue peptide, and it is a class of opioid peptide that arise from the precursor protein prodynorphin. So prodynorphin is the precursor of Dynorphin A. So when prodynorphin is cleaved during the processing by protein, it converts into multiple active peptides, and these peptides are Dynorphin A, Dynorphin B, and alpha beta neoendorphin. So by the cleavage of this peptide, we get Dynorphin A, Dynorphin B, and alpha beta neoendorphin. Now coming to the next class is Endomorphins. So Endomorphins, it exists in two forms, Endomorphins 1 and Endomorphins 2. Endomorphins 1 has a sequence of tryptophan, proline, tryptophan, phe, and Endomorphin 2 has a tryptophan, proline, phe, and phe. Again, it is a group of indigenous opioid peptide and it consists of Endomorphin 1 and 2. They are tetrapeptides with the highest known activity and selectivity for the mu opioid receptor. So as I said, it acts on mu opioid receptor. Endomorphin 1 is found in the nucleus of the salivary tract and the dorsomedical hypothalamus. It is supposed to be that endomorphins are the cleavage product of a larger precursor, as I discussed earlier in the other form of peptide, but this polypeptide or protein has not yet been identified. So still, the cleavage product or endomorphin cleavage product is still unknown. The next class of opioid peptide is actually food peptide. These opioid food peptides have length of typically 4 to 8 amino acids. And again, it is body's own peptide are generally much longer. Opioid food peptides, there are many types of opioid food peptides. One is called Casomorphin, which is isolated from milk. The other one is Gluten Exorphin. It is actually isolated from gluten. Then Gliadorphin, again from gluten, and Rubiscolin. which is from spinach. As you can see, there is a list of different opioid food peptide which has been isolated from different food product and from different food proteins. So let us first see Casomorphin. Casomorphin, again, as I said, it is one of the opioid compound. It is formed in our stomach when we drink milk. So basically it is coming out of milk. And then it is associated with the apnea, infant apnea. You might have heard of the terminology, infant apnea. And this infant apnea is actually, Casomorphin has an effect on infant apnea. And with the release of the opioid peptide, this apnea can be treated well. Gluten Exorphin. It is again a class of opioid peptide, opioid food peptide formed during the digestion of the gluten protein. So it is again formed in the body, and it has been hypothesized that people with autism and schizophrenia have abnormal leakage from the gut of these compounds. So when these compounds actually leak from the gut, then they have this problem which is called autism and schizophrenia. So that means this peptide is having an effect and having a problem with the autism and schizophrenia. Again, this gluten exorphin is of 4 types, gluten exorphin A5, B4, B5 and C. Rubiscolin. Rubiscolin is actually a class of opioid peptide that are formed during the digestion of the ribulose, biphosphate, carboxylase, oxygenase protein from spinach leaf. So as the name says that this is isolated from spinach leaves. It mimics the effect of opiates and therefore influence the brain. These peptides have much in common with the better known gluten endorphins. Again, there are 2 types of rubiscolin with known structure, rubiscolin 5 and rubiscolin 6. Rubiscolin 5 is 5 residue peptide and rubiscolin 6 is 6 residue peptide. Now, there is another class which is called amphibian opioid peptides. So as you can see by the name that these peptides are isolated from amphibians. So there are 2 types of amphibian opioid peptides. One is called deltatorphin and deltatorphin, also known as the deltorphin. And deltorphin A and dermorphin is a naturally occurring exogenous peptide, heptapeptide. So as you can say that this is like a heptapeptide, but the important part is that the methionine residue in this peptide, in this sequence, is actually D-methionine. So, along with the other deltorphins, such as deltorphin 1 and deltorphin 2, the deltorphin is endogenous to frogs of the genus Phyllomidas. Dermorphins is actually again a heptapeptide, and it is first isolated from the skin of South American frogs belonging to the genus Phyllomidas. Is about 30 to 40 times more potent than morphine, but theoretically may be less likely to produce drug tolerance and addiction due to its high potency. So, as I discussed, the other form of opioid peptides are exogenous opioid peptides, and exogenous again are 2 types, semi-synthetic opioids and synthetic opioids. So, semi-synthetic opioids are derived from opium, oxycodone, hydromorphone, buprenorphine, heroin, the most important was the heroin. These are the semi-synthetic opioids which are derived from the natural opioid peptides. And then comes the synthetic opioid, which is man-made. Man-made, I mean to say that these are synthesized chemically in the laboratory, in the pharmaceutical company, or in any such research environment. And some of them are fentanyl, methadone, meperidine, and tramadol. What is semi-synthetic peptide? The semi-synthetic opioid peptide is actually, they start with a substance that occurs in the poppy plant to make a semi-synthetic peptides. What it means that they are actually isolated from the poppy plant, from the opium, and then they are modified based on the requirement. So some of the semi-synthetic opioid peptides I would like to mention here. One is buprenorphine. So buprenorphine is a semi-synthetic opioid peptide, and it is derived from the thebaine, which is natural opiate that occurs in the poppy plant. Although buprenorphine is technically considered an opioid, it is only partial opioid agonist. That means it has maximal effects are less than those of full agonist, like heroin and methadone. The next class is Hydromorphone. Hydromorphone, as I said, it's again is a semi-synthetic peptide, and it is most commonly referred as Dynorphin, one of its brand name, because brand has synthesized this semi-opioid peptide. So next semi-synthetic opioid peptide is oxymorphone. This peptide is actually octagonal in shape, and this is where oxymorphone, because it is octagonal, oxymorphone, it gets its name, and it is the most common street name. The next semi-synthetic opioid peptide is hydrocodone. Hydrocodone is prescribed most often as Vicodin, also containing the acetaminophen. Is the most frequently prescribed opioid in the United States, and also it is synthesized from the codeine, which occurs in the poppy plant naturally, like thebaine, morphine, and opium. So again, it is derived from the opium plant. Then comes the oxycodone. Oxycodone, again, it is the most recognizable prescription opioid. Oxycodone is manufactured by modifying thebaine. Often it is also called oxycodone. There are many names to oxycodone. Then last but not the least is the heroin. I'm sure all of you must have heard about heroin. Heroin was actually synthesized from morphine in 1874 by the pharmaceutical company Bayer, and was touted as a safer non-addictive form of morphine. But today we know that heroin is to be one of the most addictive and dangerous illicit drug in existence. Now coming to the synthetic opioid, which is again another form of exogenous opioid peptide. So there are some synthetic opioids which have been synthesized purely by chemical reactions, and the one of them is meperidine. Meperidine is actually commonly known by its brand name, which is Dermol. Meperidine is used to relieve moderate to severe pain. Then one of the most used synthetic opioid peptide is fentanyl. Fentanyl is particularly potent synthetic opioid that should only be used by those individuals who are already tolerant to narcotics. And then there is next one, which is called methadone. Methadone, it was again produced in 1930 by a team of German scientists who were searching for a pain-killing drug, again which is analgesic drug, that would not be as addictive as morphine. And that is how, keeping this in mind, methadone was synthesized. Despite the fact that methadone is often used to treat the opioid addiction, it can still be addictive itself when not used correctly, which is why it is abused by many individuals. So, coming to the importance of synthetic opioid peptide, one of the most important fact about the synthetic opioid peptide is you can change the conformation, and the conformation, as I already discussed, that conformation is directly proportional to the activity of the peptide. Because we can modulate the conformation, these synthetic opioid peptides can be of much more use. And also it easily fits into enzyme. So biological degradation, facile, and because it easily fits into the enzyme activity or medicinal value, it increases the medicinal value. Also, different conformational structures available for binding, transduction, and reversal at receptors. So these are some of the important facts about the synthetic opioid peptide, why we opt for synthetic opioid peptide or sometimes a semi-synthetic opioid peptide. What is the take-home message? The take-home message is synthetic and semi-synthetic opioid peptide can be both beneficial as well as dangerous. It depends how it is prescribed and how much quantity it is taken. So that is the reason we always suggest that before taking any anti-pain tablets or any kind of opioid peptide, you should always discuss with the doctors. These drugs were all manufactured for a reason, usually to create a better, less addictive or safer painkiller than those which already existed. So students, I think I have been able to explain you well about the opioid peptide and their therapeutic use and their therapeutic importance. So now, if you look at the research which is being done in the opioid peptide field, it is completely progressing at a very rapid rate. Like most of the synthetic peptide which is being synthesized is by mostly by solid phase peptide synthesis, and by the manipulation at the different residue, as well as manipulation by different unusual amino acids. You can actually fine tune the sequence, you can fine tune the activity, you can fine tune the conformation, which will be important for the activity, which will be important for the quality and quality and structure activity relationship. So these are very important field which is actually progressing well in the field of opioid peptides. So now we may look forward to still more exciting discoveries. I mean, already there are many peptides which are in the market, semi-synthetic, synthetic, as I mentioned already, but there are many more to come, because there are many more methods which is being developed and which is being optimized for use. And by using these methods, we may be able to, synthesize some opioid peptides which will have therapeutic use, as well as these peptides will not have like side effect as most of the narcotics which we know already have. So we will try to avoid the side effect of the narcotics, as at the same time we will try to develop some opioid peptide therapeutics which will have much more activity as compared to the naturally occurring opioid peptide. Now, as I mentioned, peptides have several advantages as a potential drug, not only in terms of, like opioid peptide, even in terms of any potential drug, peptide has a very many advantages. First of all, it includes high activity, high specificity. They are very specific to specific receptors. They have very low toxicity, because these are synthesized from natural amino acids. These natural amino acids will not have toxicity in the body, and they have minimization of drug-drug interaction, so they will not interact with each other. Also, the other important factor about the peptide drug is that because these are chemically synthesized, I'm talking about the chemical synthesis, these are chemically synthesized, they cannot be cleaved by the proteolase enzymes which is present in the body, because they will not recognize as the natural or peptide which are synthesized within the body. Also, the metabolic stability of these peptides can be fine-tuned by the choices and the number of modifications induced. As I already mentioned, you can fine-tune the structure, you can fine-tune the sequence, you can fine-tune the activity by replacing amino acids, so either one amino acids or more amino acids, by the unusual amino acids, by the D amino acids. There are many more modifications which can be done while fine-tuning the activity conformation of these peptide drug molecules as well as opioid peptide molecules.